Okay, so this is the easy part. And at this point, we have our image which is a representation of some slab of tissue, right? The thickness, of course, is a function of the combination of the range of frequencies that we transmit and the slope of the gradient magnetic field. But if we measure that signal after using this slice selection gradient, and I go back and ask you the same question, right? The patient is sitting here in the scanner, and we've selected a certain slice, let's say through the chest. So we measure the signal, and I now ask you, right, here is our signal intensity, right, transverse magnetization, going away with T2 star, and we measure it at some time TE. So when I measure that signal, where in this slice does it come from? Right? We know that it only comes from that slice because we've used slice selection, as we just described it, to make sure that that's the case. But I have absolutely no way of telling you where in this slice that signal comes from, other than that it comes from everywhere. Right? And in fact, this slice is an aggregate of whatever is within the slice. Everything altogether is expressed in this aggregate signal. What we have to do now is take this signal and split it up and determine which components of that signal, right, that thing that we looked at earlier, come from which locations in the image. All right? So this is the approach that we're going to take. If we look at what has happened in order to get to the point where we can actually measure the signal, we just sort of draw a little timeline. Okay? We've had to turn on our radio frequency pulse, and at some time later on, called TE, we sample the signal. In order to select this slice, we have to turn on a gradient magnetic field at the time that we transmit this RF. And if that wasn't clear, this is an essential point. Slice selection only works if the gradient magnetic field you're going to use to select your slice is on while you transmit the RF. Otherwise, it has no effect. So we turn on this gradient magnetic field, transmit the RF. While the RF is on, only the spins in the slice are precessing with a frequency that matches the frequency of this RF pulse. Therefore, only those spins become excited. At this point in time, we've turned off our RF, we've turned off this slice select gradient, and we're basically not doing anything to the patient. We're just watching the signal decay until we decide to sample it. Okay? At this point in time, what is the frequency of precession of the spins in the slice? You know, on the left compared to the right, top compared to the bottom, it's the same, right? The entire slice at this point is only experiencing the static magnetic field strength B naught. The gradient has been turned off. So everything that is giving us signal, right, this signal here, is expressed by spins that all precess at exactly the same frequency. So what we're going to do now is turn on a gradient magnetic field which runs along one of the two in-plane dimensions of the slice. Okay? So this gradient magnetic field is causing the magnetic field strength to change, in this example, from left to right in a linear fashion. If I turn that gradient on at the time that I'm sampling the signal, that means that all of the spins in that slice, which during this period of time were precessing at the Larmor frequency, all of a sudden, during this period of time that that gradient is on, are precessing at a frequency that depends on the static magnetic field plus however much gradient magnetic field they are experiencing. And the amount of gradient magnetic field experienced by those spins depends on their location, left to right in the image. So spins farther to the left might be precessing slower than spins farther to the right. Not only that, but there is a linear progression of frequency across this dimension. So when I sample the signal at this time TE, this signal is, now there's only one signal. I have a single coil that measures the summation of all of this information. But realize that now this signal is composed of a whole bunch of different component signals, each of which has a different frequency, right? So there is, first of all, different amounts of signal amplitude at different locations in the image, simply depending on what kind of tissue is there. That's what we're trying to figure out, right? If there's, heart and lung and liver, those all have different amounts of signal amplitude. And all we're trying to do is to put it in the right place. So the reality is that there are different components of signal amplitude, and each of those amplitudes have a unique frequency depending on the location from left to right. Yet when we detect the signal with our single receiver coil, right, all we have is this one big coil here to sample it with. We detect this as one composite signal, which is essentially a sum of all of these components. So embedded in that signal is all of that information, which relates to the spatial distribution of signal along one more dimension in the image. Okay? So this, by the way, is based on this idea, which some of you may be familiar with, probably most of you are familiar with, is that if we have let's say two different sine functions. Right, this goes back to some math. If we take at each point along here the sum of each of the two components, so both functions are 0 at this point. At this point, let's say we have this much and this much, right? So that's going to put us about here. At this point we have this much and this much is going to put us about here. Here we have this, but then we have to take away this one, so we'll be at this point. This one we're at 0. This one we're about over here. Oh, color changed. And then if we connect the dots here, we end up with some complex function which is a sum of those two sine waves. So this is what's going on in this example, right? At each location, left to right, there is an amount of signal amplitude processing at some frequency. Yet when we sample the signal, we are sampling the sum of all of those components. So the signal that we now detect is spatially encoded based on its frequency. And in fact, we will call this the frequency encoding gradient. Right? By having this gradient magnetic field on at the time that we sample the signal, and that's essential. It has to be on while we are sampling the signal. As soon as we turn the gradient magnetic field off, everything sees only B naught and processes at exactly the same frequency. There's no spatial information. But if this gradient magnetic field is on while we sample the signal, then all of that spatial information is embedded in this single signal. And if we only had a way to extract that information, we would be able to localize signal along this dimension of the image. Right? And that's exactly what we're going to do next. Okay? So this process over here where I added together these two sine waves to construct this complex composite function. Does anyone know what that's called? Anyone? Well, it's a reverse Fourier transform. Okay? Adding up sine waves, just like everyone must have done this at some point, right, in some math class. This is called a reverse, well, we'll be consistent, right? This is called a reverse Fourier transform, okay? The Fourier transform that we're used to, which we're going to deal with, starts with the green and goes in the other direction. Okay? So it is essentially able to take this complex function and determine what its components are. Now, we're not going to go into this in a lot of detail, but the nature of the Fourier transform is that we are able to probe this complex function and determine a series of component frequencies, each with a specific amplitude, that combine together to give us that net result. So this becomes very useful when we are sampling the MR signal. We have our slice, and our frequency encoding gradient is applied in this direction. So when we sample this signal over time, we will be able to take this and determine a series, right, of different frequencies, each with a unique amplitude. Since those frequencies depend on the gradient magnetic field, right, this frequency information is spatial information. Right? Frequency is location in MR. And we can take this result and localize our signal along this dimension in the image.
Is that within the slice we've selected?
This is, well, you tell me.
Yes? No.
Okay. Anyone?
Yes.
It has to be. Because the only signal we have, right, let's go back to here. We turned on our RF in the presence of a slice select gradient. The only spins that have any net transverse magnetization is the ones that were excited by this process. There is no more signal. So once we get beyond this step, we are now only dealing with the spins within the slice. Okay?
So within the slice we've selected, we also have a gradient.
So what we've now done, this is a timeline, okay, of how this procedure works. So we've turned on our RF and gradient magnetic field for slice selection at the same time. That excites the spins within the slice. We then turn everything off, okay? And now all of the spins outside the slice are just laying around, relaxing like nothing ever happened. The spins in the slice have become excited, have this transverse magnetization. Okay? At some point in time, we're going to measure how much transverse magnetization is left. During that measurement of signal, we turn on a gradient magnetic field on a direction that is perpendicular to the direction of slice selection. It is along this left-right dimension in the slice. Okay? Now keep in mind, however, it's not true to say that this gradient magnetic field is only applied to the slice. The gradient magnetic field has its effect throughout the bore of the scanner.
That's only where I see it.
But it only counts over here because we only have signal over here.
And that is a separate gradient magnetic field? I mean, this is the same field that we applied in the beginning, we just do it again, or it's separate?
Is it the same as the slice select gradient? No, it can't be. Because what I'm telling you is that we've selected this slice. To select this slice, we have to use a gradient perpendicular to the orientation of the slice. We now want to localize our spins in the plane of the slice, so we turn on one of the gradient magnetic fields that is orthogonal to that slice select gradient. Okay? Did that clarify what you were? Yeah. Okay. Any other questions at this point? Okay. So at this point, we have this complex signal, which comes from the entire slice, comes from everything within this slice, everything left, right, top, bottom, and through the thickness of this slice. The frequencies that add up to give us this signal are frequencies that are being perturbed or changed in a linear fashion along this left-right dimension of the image. And those component frequencies can actually be extracted from this composite by the Fourier transform. Okay? So why don't we stop for at this point for maybe 5-10 minutes, and we'll pick this up afterwards.
Quick question. How does
Yes.
the Fourier transform, how does it figure out how many signals are making up that composite signal?
Okay, that's what we're going to get to next.
Okay.
It has to do with the way that we
Sorry.
sample the, that's okay. It has to do with the way that we sample the signal. And we'll get to that next, but good question. Where we are at this point is that we've put the patient in the scanner, excited the slice, and then during the time that we sample the signal, we have a gradient magnetic field on across one of the in-plane dimensions. Now just to clarify one thing that someone asked me during the break. So I drew this gradient magnetic field as a flat horizontal line, which may have been a little bit deceiving. So if this makes it any better, it is a gradient magnetic field, meaning that the strength of the gradient is changing from left to right in the image. As a result, the net field strength experienced by these spins, depending on where they are along this left-right dimension, varies. Right? And since their field strength experience varies, their frequency of precession varies, which means that when we sample the signal, this is composed of a whole bunch of components, right, each at a different frequency, depending on the location left to right. And we've said that we can take that composite signal and using the Fourier transform, decompose it into those components. Right? Which then each of these has two features. One is a signal amplitude, and one is a frequency. Right? Based on the frequency, we know the location left to right in the image, because this gradient magnetic field changes in a linear fashion. So we get a whole bunch of different frequencies, and we know how to line them up left to right in that image. So the issue at hand now is, how do we do this? Right? How do we go from this one single composite signal into all of these different components? Right? Well, the Fourier transform that is used in MR because we do everything with computers, right? The Fourier transform actually predates computers. There are ways to do this with pencil and paper if you really wanted to, right? It's called the digital Fourier transform. And what that requires is that we feed the Fourier transform a series of measurements of this signal. Okay? Now, this has to do with the piece of hardware that we talked about yesterday, that is the analog to digital converter. So the signal, right, if we simply hooked a little plotter up to the voltmeter on our receiver coil, we could actually record the MR signal as a continuous analog signal, because that magnetization is continuously, that transverse magnetization is continually processing in the transverse plane. Right? To deal with this in our digital system, though, the analog to digital converter is going to essentially chop this signal up into a whole bunch of pieces of information that are collected over time. Right? So during each one of these periods of time, which we call the sampling time, or T sub s, the system records the total signal amplitude that occurred during that period of time. So what we get out of this, and we can then go and record this basically in a spreadsheet, is a series of numbers, which each one is a measure of signal amplitude. And we can write those numbers down in order that we collected them over time. Okay? And this is the digital sampling of our signal that the receiver does. That's an analog to digital conversion. Okay? By taking this signal and feeding it into the digital Fourier transform, we can get out of this let's see. Right. So by putting this through our Fourier transform, we can get another series of numbers, right, which are all measures of signal amplitude. But rather than arrayed in terms of time, they are going to be arrayed in terms of frequency. Right? So what the Fourier transform is allowing us to do is to go from what we call the time domain, right, measures of signal amplitude that correspond to points in time at which we acquired them, and convert those into measures of signal amplitude, right, that are associated with different frequencies that were components of this aggregate signal. And once we do this, we can then take those measures of signal amplitude and put them in the right location in the image. Okay? Now, this allows us then to localize signal along a second linear dimension in our image. A single linear dimension, right, in the plane of the image. So I know how much signal amplitude, let's say, comes from this location in the left-right direction. But what I can't tell you is where different components of that signal amplitude come from along the third dimension of the image. Right? So all I know is that it comes from a given location left to right, but as far as top to bottom, all of that signal comes from everywhere in that top to bottom distribution. Right? This is called a one-dimensional Fourier transform. And this could be used to extract very crude information from an image. So for example, if we have a patient and we select a slice through the knees. Right? Turn on our slice select gradient in the z direction and transmit the correct range of frequencies with our RF to select this location. We now have information that is coming from an entire slice. Yet when we look at that slice, we have no idea, right, where in the slice individual components of that signal might come from. If we turn on a gradient magnetic field in this direction, right? So we have applied our RF in the presence of a slice selection gradient. And then over here at TE, if we turn on a frequency encoding gradient, we then get a signal, which the signal looks like whatever it looks like. We can acquire multiple samples of that signal over time. And by performing a Fourier transform, we can now have multiple samples of signal that correspond to different frequencies. If we simply now plot signal intensity versus frequency, what's it going to look like? What does that represent? Right? This is through the legs, right? So assuming the person's legs weren't exactly together, there's no signal outside, right? There's no signal between their knees, and there is maximal signal in the center where the tissue is the thickest. As you know, it's a round structure. It gets thinner to the sides, so the signal trails off here. So this tells us information about the spatial distribution of signal along a single linear dimension. But all it tells us is that in the center of this leg, we have this much signal amplitude. We have no way of knowing at this point where different amounts of that signal come from, even though certainly some of it comes from bone, and some of it comes from muscle, and some of it comes from cartilage, and whatever else. Right? That's the task of our next step. Okay. So an example that I have in the notes is, and that example is really there just because people usually ask this question, if we would stop now and make an image. So my image is some array of 256 by 256 pixels. And I say, I'm tired, I don't want to go on anymore. Let's just make an image with the information that we have. So what would it look like? Right? So you have this sort of box of bars. You would be forced to basically say that, well, I know that at this location, right, this is the person's, the legs that we can't really see are over here. So all we could say is that we know that at this location there's a maximal signal amplitude. But we would have to make that signal amplitude the same all the way from top to bottom, because there's no way for us to tell, right, where the signal comes from in that direction.